we are going to look on the topic target identification in drug design. This is the very important step in this drug discovery aspect since it determines everything that comes after during the designing and development and post-development phases. So let's see a short intro before we going into the approaches. So this is the timeline we usually have seen for drug development. So in this diagram, we can see that where the target identification exactly happens. So it is the first step. So after that, there are 7 to 10 years of work, which cost around millions of dollars to find one promising drug. And so then comes the clinical trials of preclinical, clinical testings, which takes about 1 to 2 years, and another millions of cost of investment and clinical testing. So that again divides into phase one, phase two. So that doesn't stop with that. Again, it goes into the approval part. So FDA, Food and Drug Administration, EMA, European Union, so they all approve it. And also after the approval, post pharmacovigilance process will go on to make sure that. So the entire pipeline will takes around 13 to 22 years, where the initial part alone takes around 7 to 10 years. So, so these whole pipeline start in the target identification, the basic foundation. So if we choose a wrong target, that is not actually involved in a disease, and it will collapse everything. So target identification is the very critical steps. It determines the success and failure of the entire project. So today what we are going to see is like we understand the target identification fundamentals and the prediction methods, druggability assessment, target validation, and multiomics data analysis and clinical relevance as assessment. So we see a short history. So how this target identification started. So in the earlier 1900 to 1080, so earlier era, so it was like a trial and error method. So they would take a natural products like plants, fungi, bacteria from soil and screen them, and like they expose to the organisms and bacteria so to see what happened. And their problem was like they had no idea how it worked and why, like what was the target and all. So after that, 1980s to 1990s, so there comes the mechanism-based target selection. So what exactly happened is the molecular biology revolution happened. So the introduction of recombinant DNA technology through gene cloning, and they could isolate product-specific disease-causing genes, and then it progressed to the structure. So the entire strategy shifted towards it. So there comes the structure solid techniques like X-ray crystallography and protein purification techniques and all. So then after this preliminary things, the entire was shifted to the genomics era in 2000. So there comes the uh, like how to say the human genome project. And between 2003 and 2025, human genome project was successfully completed. So after that, nearly 20,000 human genomes are solved. So we can got the entire DNA sequences. So that was a very big breakthrough. So that we can got that gene expression microarrays, and from the gene expression we got proteomics, and they derived the systems biology approach. So everything was derived from the human genome project. Then later it was again shifted to the computational era, combined with the artificial intelligence introduction. So there comes all the computational tools and machine learning approaches they have used, and in current integrated multimodal approaches have been implemented like AlphaFold, AI/ML predicting, and then it deviates to the personalized medicine, off-target analysis, and also multi-target drug design approach. So it becomes from the it has started from trial and error method to rational design and high throughput screening to artificial machine learning methods, then to integration of these multimodal approaches. So why we need target? Why we need to identify potential target? Because there are four important points in the it de-risks the project investment and timeline. So once if we identify very wrong targets, then it becomes a failure, and it like no matter how good our chemistry is. So it is very important, and then enables rational drug discovery and selectivity. So once a identified target is validated, and we get the three-dimensional structure of the target, so we have to design an inhibitor for it. So there comes structure-based design, which minimizes the off-target effects. And the third one is the therapeutic potential and clinical efficacy. So once we choose a target, it directly impacts the disease indication, patient population, and clinical outcomes. So those parts come in that. And then fourth point is guides personalized medicine strategies. So due to the presence of high experimental models, precision medicine requires the understanding of entire target networks, just not just the individual proteins. And there comes the network pharmacology approaches and etc. And that. So we now go on to the topic of target identification strategies. So the target identification strategies can be classified as the chemical approaches, which is the direct identification, and genetic approaches, computational approaches, and systems biology approaches. So in direct chemical approaches, there comes the again classified as using it is respect to each experimental methods by using chemical probes, affinity chromatography, isolation and identification of the target molecule, and ligands binds to the target protein directly, and using mass spectroscopy. And in case of genetic approaches, the approaches includes knockout, knockdown strategies by using loss of function and overexpression, CRISPR-Cas9 studies, which are very powerful technique, and RNA interference and genetic screens. And computational approaches, structure-based virtual screening, machine learning approaches, sequence homology-based target prediction, protein network analysis, ligand similarity searching, and in systems biology and multi approaches, we use genomics, proteomics, metabolomics, transcriptomics. So these integrative analysis, like pathway analysis and network pharmacology. So that is to identify disease-causing nodes in the biological networks, and it also reveals interconnected targets in the disease pathway. So that's how the system integrated approach is used. So let's look on to the in a chemical biology approach, which is called as a direct approach for target identification. So this is where our designed molecules becomes experimental tools to find and validate the targets in the real biological systems. So that's how it works. So we are going to find our target in the real biological systems, unlike any computational predictions or any other experimental approaches. So first, here the key factor is the chemical probe. So what makes a good chemical probe to initiate this workup? So a chemical probe is a small molecule, so which has should have a size less than 500 Daltons, and the potency less than 100 nanomolar, and greater selectivity for half targets and cell permeability. So these criteria matters to design a probe. So before starting the experiment, so we should validate that the molecular probe meets the strict standards like that. So one of the approaches is affinity-based target isolation. So once we have a validated chemical probe, we use it to physically isolate our target protein. So the workflow includes like first we label the probe and we attach it to the tags. Tags includes biotin, fluorophores, or other molecular handles, and these should not interfere with the binding, but it allows us to track and capture the probe later. So it will not affect the binding. So it just acts as a trackers. Next, we add the labeled probe into the cells or the cell lysate. So we are letting the probe to find and bind to the target proteins in the mixture, like in a biological mixture. So we should remember that there are there are thousands of proteins present inside the cell, but our selective probe should preferentially bind to our intended targets. So once we have designed this, and the next step becomes the isolation step. So the isolation steps includes the affinity chromatography and the immunoprecipitation techniques, and finally the mass spectrometry. So in affinity chromatography, the biotin tag where the on the probe binds to the strepta within beads or the other affinity matrices, pulling down whether the probe is bound to. Otherwise, we can use immunoprecipitations with the antibodies against the tag or pull down using the capture mechanisms. So once we have pulled down those targets, so we use mass spectrometry for the identification. So we digest the isolated proteins and run them in the mass spectrometer. So this mass spectrometer tells exactly which proteins are captured and with the sequences and modification information. So this is a very powerful approach because it is unbiased. So we are not assuming we know the target. So we are just asking the cell by using the chemical probe. And what does our molecule bind to, etc. So next one is the activity based protein profiling. So this this adds specificity based on the enzyme activity. So previously we pulled down based on the binding. So here it is based on the enzyme activity. So this ABVP uses mechanism-based reactive probes. So the previously it will not interfere with any mechanisms, but this one uses mechanism-based reactive probes. So instead of passive binders, these probes have electrophilic warheads, which has a reactive functional groups in it. So when the probe encounters on the active site of an enzyme, it covalently bonds to the active site. So these warheads react specifically with the nucleophilic residues, which is present in the binding site. So those residues are like serine, lysine, cysteine in the active site. So on the target specificity, so only the enzymes with the active nucleophilic residues gets labeled, and this is more than the general binding. And the next one is it is like a genome-wide off-target discovery. So in ABBP, probes are not designed to hit just one target. So use them on the whole proteome, all the protein, which means it contains thousands of proteins. So there the probe covalently labels every enzyme's active site it can access. So then we use mass spectrometry, and this becomes an unbiased proteomic analysis to identify all the proteins that we got labeled. So this how we can discover the unexpected targets and off targets systematically is without bias. So we may design a probe for one enzyme, but this technique reveals it also hits three more than our protein, like other enzymes, so that we didn't expect that. Because we better we can know this now than in the clinical trial. So it can predict off target also. So the next one is the thermal shift and binding assets. So this is more towards the biophysical measurements between the it is not a problem. We can name it as a ligand or protein bindings. So instead of in earlier, so what is in the pull down? So now we are moving on to how strong the binding is. So for this, we can use techniques like differential scanning, fluorimetry, surface plasmon resonance, isothermal calorimetry, ITC. So these are all the biophysical techniques. So in DSF, differential scanning calorimetry, the fluorescent dye binds to the exposed hydrophobic regions of the proteins. So once we heat the protein will get miniature, that is, it unfolds and the fluorescent changes. So if we have a ligand bound to the protein, it stabilizes the structure and the protein denatures at a high temperature. So we call this as a shift, thermal shift. So by measuring how much the melting temperature changes, we can quantify the ligand binding strength. So it also works on the crude samples. The next one is the SPR, surface plasmon resonance. So our target protein is immobilized on a biosensor chip. So and ligands flow over it and binds. So as mass increases on the surface, the optical properties changes. So we measure the real time. So this technique gives the association and dissociation rates and equilibrium dissociation content. So this one is the label-free technique. And the next one, ITC, so isothermal calorimetry. So this is the direct measure of heat released or absorbed when a ligands bind to the proteins. So we get the dissociation uh, dissociation constant with also the thermodynamic parameters like entropy and enthalpy. So this gives the nature of binding. So it is like driven by favorable interactions or entropy like that. So these three measures will techniques will give us the measure of the target and compound engagement. So we can have proof that our molecule actually binds to the protein or the target we think, and it uses in the validation stages. So the next one is the uh, cellular and in vivo target validation. So finally, we need to validate the targets in the physiological concept, like inside the living cells. So a compound, our screen compound may bind to a target protein in the biochemical assays, but in living cells, it may not reach the target. Maybe it get metabolized too quickly or other poor cell permeability or other parameters like that. So we should make sure whether it works inside the cell. So there comes the use of proximity dependent assays, FRAP, BRET, and split turbo ID. So this like FRAP is used on the fluorescence where the ligand is labeled with a fluorophore, and we bleach the region of cell and watch how quickly the fluorescent recovers by diffusion. So if the ligand is bound to the immobile target proteins, the recovery is slower. So this indicates the on-target binding. So then when is the BRET? So it is like bioilluminescence, resonance, energy transfer. So this we tag our target protein with a fluorophore and our ligand with the complementary fluorophore. So when they are close, that is the ligand bound to the target, the energy transfer happens between them. So it produces signal. So this quantifies and in the live cells. So the next one is the split turbo ID. So here the biotin ligase enzyme splitted. So first part is our target protein, the second part is our ligand. So when they interact, the enzyme is reconstituted and the biotin labels nearby proteins we capture them. So and then is the mm, yes. So then the next technique is using the genetic approaches. So genetic approaches, gene knockout and knockdown studies. So let's start with the CRISPR-Cas9. So this becomes this which has now revolutionized target validation. CRISPR allows us to make permanent genetic changes in data. Like we are cutting one gene from the gene. So this is different from the chemical inhibitions where in that part we are still present, that target is still present but inactive. But here the entire gene is gone. Why it's a this is a now it has been a powerful technique. Why? Because it answers the questions like if we inhibit an enzyme, so with a drug and disease phenotype improves, we can wonder, so is it really this enzyme or we are hitting with other off targets? So with CRISPR knockout, so we can identify why if a gene is deleted and if the it causes the same phenotypic change. So we are rather after when we are proven causation. So, mm, for example, how to say, for escape pathogens, they create CRISPR library and they for targeting every gene in the bacterial genomes and introduce the library into the pathogen, and they grow cells under the selective pressure. Uh, and then they sequence which the guide RNA survives. So the survivor guides tells which genes are essential for the pathogen fitness under the given specific conditions, like that. So the then one is, but it has some limitation. So some genes are so essential that a complete knockout is lethal, so making them difficult to study. So then comes the knockdown part comes. So here we use RNAi knockdown approaches that are different from the previous one. So there, instead of the permanent deletions, here uh, we reduce the protein expression. So ultimately, central dogma is the important approach, so which ends in the protein expression. So here we are not remove the gene, so we are silencing it. So for silencing, we use predominant approaches like siRNA and antisense oligonucleotide and shRNA. So siRNA is small interference RNA. So it is a double stranded RNA sequences that matches our target mRNA. So when it is introduced into the cells, the RNA interference machinery recognizes the siRNA, so finds the matching mRNA and degrades it. So once it degraded, the protein expression stops. But it is a temporary silencing. Rather in antisense oligonucleotide, so single stranded DNA or RNA sequences that are complement to our target mRNA is designed. So they bind to the mRNA, so preventing the translation. And in case of shRNA, which is a short hairpin RNA, similar principle is used. But here the hairpin is encoded by the DNA, which is usually like a vector and expressed inside the cells as an RNA. So the cell processes it into siRNA-like molecules because it is encoded. So we can have a long-lasting knockdown as long as the plasmid vector process inside the cell. So why we use knockdown instead of knockout? So some genes are absolutely essential because we cannot knock out everything. So complete knockout is embryonically lethal and cause immediate cell death. But the partial knockdown, reducing the protein to certain percentage, that might produce a disease phenotype without being lethal to it. So this is why we study genes that are too important to delete completely. And also in case of uh, dose dependent responses. So with in case of knockdown, we can study the dose response relationship. So reduces the protein to 80% normal and no phenotype. So we can reduce to 50% that mild growth is effect. So reduce to 20% severe phenotype. So this dose dependent, it's strong evidence of causation and tells us how much inhibition is needed therapeutically. So, and in case of next we go to the case of transcriptomics. So let us shift to the perspective. So instead of looking onto the one target, let's look at the entire transcriptome. So where the all gene is being expressed. So microRNA and RNA-seq technologies measure the expression level of every gene inside a cell under specific conditions. So we can compare infected versus uninfected cells and diseased tissue versus a healthy tissue and treated versus untreated cells and virulent strain versus attenuated strain. So um, what we can infer from this is this produces a big data, so massive data, because thousands of genes, so each with an expression level. So again, we will have a questions. Okay, we have a expression level, but which genes are differentially expressed between the disease conditions? So there comes the question. Using the computational analysis, we can get the answer for this. So here, the computational tools again combined with this, like CADD pathway enrichment, gene ontology, and network analysis. So we again see this in the computational part. So this transcriptomic studies is in combination with the computational predictions we should do. And then network pharmacology, so network and systems biology. So here, the concept is most diseases aren't caused by a single proteins. So they can be caused by a network dysfunction. So here comes the concept of protein-protein interaction databases. So databases like string intact we can use. So string is search tool for retrieval of interacting genes and proteins. So an intact which focus on experimentally validated interactions from the literature. So this database queued these data and it helps us to bind the interaction networks. So where where this network pharmacology concept comes is like it from the traditional drug discovery focus, which focuses on a single target. But proteins exist as a networks. A protein might interact with either but one protein, two proteins, or it forms a network. So, but if we inhibit it, one protein, we do not just affect one protein. So, if it had forms a network, we perturb an entire network. So this might be a problem, like it causes a problem of off-target effects. And so in network pharmacology, we target a hub protein and network bottlenecks proteins that are central to disease-driving pathways. So how we can identify the hubs, and there are also a set of analysis for this. So co-expression analysis, degree centrality, between a centrality. So these are the analysis we can use. So in co-expression, what we can infer is if a gene are consistently expressed together across many conditions, like their end product protein is function together. So genes with co-expression patterns may have connectivity in our hubs. And degree centrality, so in a network, so in a protein network, highly connected proteins are considered as hubs. And between a centralities, proteins that lie on many shortest paths between other proteins or the bottlenecks. So this is how we can predict it. And next one is phenotype to genotype mapping. So um, in model organisms like zebrafish, so we do mutagenesis then screen for the mutants with disease-relevant phenotypes. So if we want to understand mechanisms of innate immunity, so we mutate the zebrafish and screen for the larvae with impaired immune response to the infection. So why we use this means, so we are not assuming which genes are essentials. We are letting the organisms to tell us. So these genes, when knocked out, cause a phenotypic change in data. So for that, in case of humans, genome-wide association studies are used. So patients with the disease are looking for genetic variations and variants significantly associated with the disease susceptibility identified. So these variants we can often map with the involved infections and immune responses or other pathogen survival. So these genes are nominated therapeutically and relevant to targets. So proteome, next comes the powerful topic called proteome-based stable isotope labeling. So these are the methods associated with the proteome-based studies. So it is an approach for quantifying the protein changes. So we grow cells in two conditions. For example, uh, drug treated versus control. So if we culture each media with heavily labeled amino acids, isotopes but not radioactive. So when the cells incorporate these amino acids into the proteins, so these proteins become slightly heavier. And the workflow includes like labeling, collection, pooling, and mass spectrometry analysis. So the drug treated cells grow in the heavy medium and control cells grow in the light medium. So we extract, then after that, we extract the proteins from both conditions and we have to pool them and run through the mass spectrometry. So in the mass spectrum MS, so we can distinguish the heavy labeled peptides from the light labeled peptides based on their mass to change ratio. So heavy peptides are slightly heavier, so they separate from the light peptides. So from this, what we can infer is proteins that are directly labeled by the drug might be degraded, and proteins are upregulated in response to the drug treatment and downstream signaling and both direct and indirect effects. So in this figure, we can infer that these two strategies. So like in ICAT labeling strategies, uh, they have two labeling and then quantification. So in uh, eye track. So this again terms as isobaric tag for relative and absolute quantification. So apart from previous technique, here we use different labeling strategy. So instead of the isotopic labeling, so we chemically label the peptides after the protein extraction. So the flow looks like protein extraction from the samples, so multiple samples. For example, disease versus healthy and treated versus control and digest into peptides, then labeling. So each sample gets labeled with different isobaric tags. So these are chemical labels, not isotopes. And then again goes to the mass spectrometry. So during the mass spectrometry, the isobaric tags fragments differentially. Actually, they release the reporter ions with a different mass to charge ratio. So this how it occurs. So what we can infer from this technique is the fold change and quantity and whether the proteins are dysregulated and whether proteins whose abundance changes upon the drug treatment. So the quantification and disease signatures and target engagement. Then comes the AZMS. So, um, so uh, here we use the drug compound itself as a probe instead of other isotopes and probes and all. We use our compound as a probe. So we immobilize the drug on beads of chromatographic column and we add our lysate, which consists of all the proteins, and we capture. So the proteins bind to the drug in the column, and we wash the unbound proteins, and we elute the bound proteins, and it is analyzed by the LCMS. So here we use LCMS because it is like it is liquid chromatography, which separates the protein mixture, and tandem mass spectrometry, which identify the each protein. So it combination of it. And the mass spectrometer fragments each peptide and matches the fragmentation pattern to the known sequences. So at final, we know exactly which protein bound to our drug. So what we can infer from this technique is known targets and unexpected targets, off-target liabilities, and tissue-specific targets, since we use differential cell types. And moving on to the TPP and DARTs. So this is TPP stands for thermal protein profiling, and DART stands for drug affinity responsive target stability. So here we heat or use a heat or protease to denature the proteins, then measure which proteins are protected by the drug binding. So we denature and we use drugs. So in TPP, the flow is like the principle is when a small molecule binds to the protein, so it stabilizes the protein's structure. And during experiments, so heat the cell lysates to increasing the temperature and denature it, and protection. So if a drug is bound to a protein, it stabilizes the proteins, otherwise it denatures. So quantification, so we use again quantification techniques like we previously saw ITRAC and SILAC. So in DARTs, same concept, but we add a protease to the cell lysate, digest it, and again we give a drug uh, and proteins with the drug bound are protected from the digestion. And again, we measure with the mass spectrometry. So drug bound proteins are protected and survive longer. So unbound proteins are digested faster. So why we can use this is means no prior biochemical knowledge is needed in case we can use this technique. So the next one is the unbiased proteome Y discovery. So this is like an integration of each techniques we have previously seen. So uh, for mm, in SILAC, what proteins change in abundance response to the drug, and in ACMS, what the proteins physically bind to the drug, and in thermal assays, so whether the proteins are thermally stabilized by the drugs. And it is we can get a complete target engagement profile with using all these techniques. So from that we can get a mechanism of action. So complete pathway from the drug binding and protein changes to the phenotypic effect we can change. So next we moving on to the computational target identification. So here, okay, wait a minute. I think I cannot remove this.
What happened, Srimati?
Ah, no, no. I just trying to remove that line from this. Okay, sorry. So okay, now we move on to the computational approaches. So far, we have seen the experimental approaches for target identification from the protein sequences and DNA sequences. Now we use computational methods. So the very first methods we come is a homology approach. So why we use this computation methods means again, so the bioinformatics pipeline will provide us a very uh, genome-wide approaches to prioritize our targets, and uh, we can screen more targets rather we use the experimental approaches. So um what condition or what criteria used in the homology is like if a drug target works in one pathogen, so the orthologous protein of the same protein in other pathogens also can be considered as a candidate. So in that criteria, we use the homology based approaches. So for that, we use BLAST based, like the BLAST. So basic local alignment search tool. So it searches for the homologs based on the sequence similarity, and PSI-BLAST. So PSI-BLAST is a position-specific iterated. So it can gives us the sequences which was evolutionary distant but biologically relevant. And then and the advanced one is the profile-based searches. We use tools like HMMR or JackHammer, so which compares the query sequences against the HMM is hidden Markov models with the concept of the protein families. So for each search, there is a certain similarity threshold values to be considered. So, for example, in PSI-BLAST, it can detect homologous that share the same evolutionary ancestors. So even when the overall sequence identity is very low, it can find out the evolutionary related proteins. And then in case of functional, so once we have identify the sequences, then we should infer whether it is functionally similar or what is the functional parameter. So beyond the sequence similarity, so do these multiple proteins share the same functional domain or the catalytic motives? So if they share means, then they likely to overlap the uh, binding site similarities, which can be inhibited by the same molecule. So For that, the databases like PFARM, Interpro, and SMART can be used. So in PFARM, which contains more than 19,000 protein families, and it is a manually created data. And then Interpro, so this integrates PFARM, SMART, and other domain means protein domain specific databases. And SMART, so it particularly focuses on the protein domain conservation and the architecture. So then pathway analysis and metabolic reconstruction. So once the identifying the targets from sequence, then functional domains is complete. So we should know that where these identified targets comes in the cells. So whether these targets fit with any networks or pathways, metabolic pathways or regulatory pathways or any other essential cellular processes. So where this happens, where this proteins goes and works. So this is revealed by the network analysis. So the key databases used for it are the CADD databases, Reactome and String. So in CAG, they have in the databases, they have more metabolic pathways and regulatory networks and disease processes. So in Reactome, it has human and organism specific pathway interactions, and String is like a protein-protein interaction, so which is based on the structure, sequence, and literature data. So next comes the expression network. So genes or proteins work together, which have similar expression patterns across the conditions and physical protein-protein interactions or the genetic interactions like that. So we use the principle like guilt by association. So we have one validated drug target and we can get its immediate network neighbor. So protein that directly interact or co-express with that. So why we have to do it and what we can get from this is if a protein physically interacts with our known target and they likely function in the same molecular way. And if a gene has a co-expression between it, and they are likely to be function in the same biological outcome. And if mutation in one gene or synthetically lethal with mutations in another gene, so that targeting both creates a powerful effect. So, and next is the systems biological level approach. So, in previous approaches, each provided each information we could get. So, this is where the integration happens. We combine sequence homology, pathway, and druggability prediction and network centrality. So, into a unified mission. So, what we are going to integrate here is for each protein, we get how conserved the protein is. Like example, if we take an escape organism and we have a target A, whether the target A protein is conserved in all the organisms, whether that particular protein is essential in all, so that we can take that one particular protein which is important in the organism. And pathway essentiality score. So if we what happens if we remove the protein? So if the pathway is blocked, if the block means it becomes an essential, so or compensated or the pathway from pathway plus analysis. So there comes the use of other databases, so DEG and PEG, so databases of essential genes and proteins of essential genes, and then the druggability score. So whether the target protein have the known binding pockets and whether it is suitable for the small molecule inhibition. So this information we get from the structure databases and druggability predictors. So, and then comes the network centrality. So is the protein is the hub in the network of proteins. So this we can get from the string and network analysis algorithms and then expression profile again. So from the transcriptomic databases. So these features are integrated either integrated as a single step or can be converted into a machine learning model. So next one is the inverse virtual screening. So this is a very small concept where we take a single compound and screen it against the library of protein structures. So this approach helps us to identify the off target. So that is another approach, or that is another we will see about the off target. So off targets are the one which those unintended the binding partners. So that may cause side effects or toxicity. So this can be useful to find we want to develop any polypharmacology or in case of to predict the off targets. So then protein protein interaction network. Uh, if we take an insider cell, protein will not act alone. So they physically interact with the other proteins as a partners and forms a complex of interactions. So we have to mark them using the databases. So there are many protein-protein interaction databases are available, string, inact, and biogrid. So what are the essential parameters we can infer from those databases are the nodes and edges. So where the nodes are act as a proteins and in edges are interactions between the proteins. So once the network is built, so from the graph we have to infer the hub proteins, which are highly connected nodes with many interacting partners, and the bottlenecks are nodes that lie on many shortest paths and control the information flow like that. So mm, then comes network pharmacology, so which connects the compounds, targets, and diseases as a framework of network. So here, network topology, we build a multi-layer network. So, for example, one layer for chemical compounds and the next layer of protein targets for disease phenotypes. So, and the edges are and the network is connected by the edges, which represent the compound binding to the target and target associated with the disease and functional relationship between this. So what we can get from this network pharmacology is we can predict new targets of known compounds, and we can get the polypharmacology. So when a compound binds to the multiple interconnected proteins, so that comes as the polypharmacology. So we can understand how a single compound can influence the entire pathway or modules. So the key idea is, so if a compound hits multiple proteins that are functionally connected to the network, the result can be in a synergetic disruption. So we are not just turning off one step, we are perturbing a whole disease module. So the next one is the systems level pathway analysis. So we know that proteins involved in the metabolic and signaling pathways. So we can again use databases like CADD Reactome to predict this. So we can place our targets into a pathway diagram and we can check so where exactly the pathway does this target occurs, and whether it is like the pathway is linear or the pathway is branched or again it connect to the another network. So and what happens in the upstream or downstream if we inhibit particular target. So what what we predict from this is we can predict the phenotypic consequences and identify essential dependencies and detect the potential bypass mechanisms. So we can aim at different points in a pathway to prevent the rerouting of it. So we can also combine enzymes and nodes from the pathway. So the next one is the off-target analysis and selectivity prediction. This is another most important off-target prediction one. So hmm, one compound will likely to bind with more than one protein target. So some of these will be desired as targets and other are off targets, so that can cause side effects. So computationally, we can predict off targets using the structural similarity and docking and pharmacophore models and machine learning approaches, and then map those predicted off targets to the human. Human means we can use protein-protein interactor networks, and in an off target, if an off target is lies in an essential human pathway or is an hub in any critical signaling pathways, then even a weak binding might induce the toxicity. So off target prediction is important for our data. And then comes allosteric site prediction and regulatory networks. So so far we focused on only the active site, which is an allosteric site. So there is another category allosteric site. So proteins can also be regulated by the allosteric ligands, and regulatory proteins are cofactors and post-translation modification. So this network analysis can also uncover the proteins that regulate the function or expression of our primary target and allosteric modulators. So nodes which are perturbed, so modulate the activity of multiple downstream. So the targeting the allosteric site, it also has several advantage. So what we can infer from this is allosteric modulators. So we can identify the allosteric modulators for our target protein. And the next one is the sequence-based target prediction. So um, so we start again, start from the blast. So we take a protein and blast it against databases like the RefSeq database, eProt, and it will give a list of proteins from the other organisms. So if a protein shares more than 40% of the sequences, and then they usually have similar 3D structures, like again rollbacks to the Anfinsen's principle. So BLAST helps us to rapidly identify the orthologous targets. So, and then another one is the sequence protein family. So here we use tools like PSI BLAST and HMMR, so which are not which have low similarity, but at a protein family wise, they are similar. So the next one is the functional annotation. So if you know the catalytic residues or the binding site of the characterized target protein, so we can transfer that information to its orthologous based sequence similarity identity. So it is based on the binding pocket. And the next one is the machine learning target prediction. So now in this era, machine learning and deep learning models have been in trend to make predictions. So, but it needs a large amount of data. So, random forest and ensemble methods are one of the methods. So, they quickly screen large library of compounds. So, and then in deep learning neural networks, so they are like a multi-layer perceptron. So, it can handle more complex data, and uh, they can get input like graph features and 3D coordinates, sending recurrent RNN, so recurrent neural networks and sequence models. They use architectures and transformers to learn from both from protein sequences and compound representations, and integrating multiple methods. So we have seen each computational methods and machine learning for predicting targets. Also, um, uh, e- computational prediction alone are not enough. So we need experimental validation also. So they're all multi-omics data integration, orthogonal experimental validation, and iterative refinement looping. So in multi-omics, we just rely on single predictions. So we should combine with the real experimental data like gene expression, proteomic data like that. And in case of orthogonal experiment, this we should make confirming our predictions usually completely independent of the experimental methods. For like we can for direct binding confirmation, you have seen ITZ and SPR to measure the binding affinity like that. And for genetic validation, we use gene knockout or knockdown approaches. And in case of proteomics, we confirm that when we inhibit the target, the predicted pathway actually get disrupted or not, like that. So in iterative refinement loop, so when we do the experiments, we get new binding affinity, structural insight, or activity. So instead of just going into the way, we feed it back to our ML model. So we retrain the model, and we get new data. So the next time we make predictions of the new diseases or new pathways, the model gets smarter. So they conflict to like either computational tool or ML prediction, experimental validation, model refinement, and better predictions. That causes like a feedback loop. So so far we have looked upon the target identification, but we have to make sure that our target is actually a druggable. So that requires structural requirements for the druggability. So most of the targets can be proteins. So the proteins need a well-defined binding packet to for a ligand binding, and they can be hydrophobic character, and it should be buried, and it is like it should be druggable. It can be either orthosteric or allosteric. So the next one is the ligand compatibility. So the target ligand should bind with our compound. So for that, there are some rules for the compounds like Lipinski rule of I like that. So the binding affinity we need must match with the real molecules. So the next one is the biological essentiality and functional aspect, functional impact. So the target must be essential for the disease, but if the knocking it out doesn't kill the pathogen or stop the disease progressions. So targets and essential pathways uh, are kind of great candidates. So non-essential targets might look druggable, but will not give the therapeutic benefit for that. The next one is the selectivity and avoidance of off targets. So the targets need to be selective enough to avoid the off target. So if it has an unique sequences or features or a binding pocket which are not found in humans, it is inherently more selective. But for targets with human counterparts, in case of the bacterial one, I'm saying, so we need careful design to minimize the side effects or often through structure-based or any other allosteric predictions. And at last, so if there are existing drugs or clinical evidence or tools for the targets, whether I mean, it is already proven as a druggable. So for novel targets, we need computational and experimental validation to show it is truly druggable. So before we invest the long-term pipeline for the drug discovery. So the next one is the structural features for the druggability. So again, the orthosteric binding pocket features and pocket volume and geometry, and then allosteric binding sense regulatory modulations and comparative structural analysis are important for this. So assessing off targets, so this is very important. So because hitting the wrong protein can be just as missing our target entirely. So for computational off-target screening, we use inverse virtual screening to check if a compound binds to any other human proteome, and we screen our compound against the structural databases like PDB and rank of the targets and by the predicted binding affinity. And the other one is the kinase activity. So this is in case for kinase inhibitors or enzyme families. So we access the selectivity across the whole kinase superfamily and related isoforms and multi-kinase inhibitors. And the next one is the cross-reactivity human ortholog. So we look for the human protein similar to our bacterial targets using sequence and structural comparison. So if a compound binds strongly to the human ortholog, then it is like leads to target toxicity. So here comes the structure-based drug designs, which helps us to know the differences between these two targets, like our target and the off targets. So next again, so one of the component is the ADMET predictions, which we had seen earlier, and then multi-parameter selectivity, hit progression. So then we move on to the target validations. Again, in target validation, so even if we start from the computational predictions, we have to again validate our targets. And in going back to the our previous chemical-based approaches, gene knockout, and RNA interference, co-immunoprecipitation, and thermal shift assays, and cell in case of cellular context. Those validation part is important, and also there are functional assay is also possible. So because we have picked our target and designed our emitter, so we have to know how it actually works. So the biochemical target exercise and enzyme kinetics. So here the concept is the enzyme kinetics. So what happens here in the biochemical target means we have to purify the proteins and measure the catalytic activity and inhibition kinetics. So those kinetics terms comes here. And in case of cell-based phenotypes, so we may remove the we move on to the cell base. So we measure the bacterial growth inhibition and reduction in the virulence markers and change in the metabolic curves, metabolic flux, and we also discover the dose response curves, etc. So, and the next one is the tissue level. So, again, we move on to the next level, like tissue level. So, here, um, we validate our targets in disease level relevant models, and mm, we measure virulence and evaluate the in vivo selectivity as an and in target pathway biomarkers, we quantify the downstream pathway biomarker and we use metabolomics, transcriptomics against, and we can assess the dose dependent biomarker, and it provides the on target validation also. So here target redundancy and epistasis analysis. So here we use combination of studies like we simultaneously inhibit multiple pathway targets. So we have to see there is a functional redundancy and we measure the synergetic effects. So functional assays are like how we move from the prediction to proof. So making sure our inhibitors actually work in the biological system. After that, once we have identified everything, so we should do the clinical relevance assessment. So genetic association studies. So we look at the genome-wide association and the target expression and tissue specificity. So again, transcriptomics and proteomics studies will come. And in the clinical validation, we search for the FDA-approved drugs or clinical compounds, and in therapeutic index and patient populations, we have to again calculate the therapeutic window and off-target toxicity like that. So in resistance mechanisms, we need to look in case of microbes. So we look at the known resistance mechanisms, and if the target mutations confers resistance, we need to address that also. So these clinical assessment is to make sure that our drug target is not just good. So it also work for at the patient level. So once, so this entire is on a theory one. So now we move on to the tools what we have to use for the drug target identification. So I will give you overview of the tools. So first, for disease mechanism mapping, so we start by gathering all the molecular data. So we can go like genomic or transcriptomics, proteomic based one, and we from disease samples, and also we can get it from the published literature. So we look for genes or proteins or pathways that are consistently altered in the diseases. So the next one is the gene identification. So we use databases like DisGNet, OMIM, or TCGA to find the genes which are linked to the diseases. And then we run enrichment analysis like GOKID to see which pathways are enriched in these disease associated ones. And once the gene has been identified, we go for the protein identification. So here in this part, we map those genes to their corresponding proteins. So map genes to the corresponding proteins using uniprotein PDB. And then we analyze protein sequence and structures to get if they are drugged or not, druggable or not. And then target prioritization. So here we filter the proteins by certain parameters like essentiality, how well it is conserved across the species. And in case of microbial targets, it should be have absence in humans. And also we also use network analysis tools to find the hub proteins in data. And then uh, we move on to the computational target identification. So here we used methods like, so far we have seen BLAST, HMM to find the homologs and orthologs, and we use structure-based methods for inverse virtual screening and then target validation. So again, so once all are designed, so we have to go for the experimental validations by using biochemical assays and screening approaches, and then integration. So we have to integrate both the results, and we use sensible scoring to prioritize the best targets. So here, so disease mechanism mapping. So uh, Here, I have listed certain tools in that. So we actually map the disease mechanism. So connect and further we use these tools. So we have to gather the molecular data. So we start by collecting the genomic, transcriptomic, and proteomic data from the public databases. And then for that we can use this OM, IM, TCGA, and GEO. So gene expression omnibuses and unexpressed. So then we move on to the identify the disease associated genes. So once we have this data, we use OMIM to connect specific genes to the known genetic disorders or confirming the relevance to our diseases. So we also check in Jivas also to find the genes that are linked to the genetic diseases. But in that we link the disease to genome-wide association, and this helps to get which genes are mostly or strongly associated with it, with the disease susceptibility. So once we have gathered the molecular data and we have got the uh, associated genes with it, we have to map the genes with the protein. So that is the next step. So for that, we use the INIPRO databases to get the protein sequences. So this gives us the detailed information about the protein sequence. It also have the information about the structure and function. And also we use literature and pathway context. So we use for literature, we use PubMed to search for the published literature, and so that on the genes and proteins. And the next one is the gene identification. So once we have identified the genes or proteins, or again, this is only particular for the genes identifiers. So this we have to find the disease associated genes. So we have to use databases like GeneCast, HGNC, and David and Richard. So this what we can get from the diseases. This is, means we use them. This will pull together the genetic evidences from multiple studies so that we can see which gene pop up most in the frequency. So in case of enrichment analysis, so what is meant by the enrichment analysis is once we have a list of genes, instead of one gene, if we have a list of genes, we want to know which pathway are involved in it. So that's where the enrichment concept comes. So we use tools like the David, Enricher, G Profiler, Panther to find the pathways that are enriched with our disease associated genes. So these tools helps us to connect the dots. So like showing us the biological process and the pathways are most affected with diseases. So then pathway annotation. So once we have completed the gene set enrichments, again, so we also curated the pathway databases like the Reactome and CADD. So to get the detailed pathway annotation. So these resources gives us about how our genes fit into the known disease pathways. And gene information. So for our particular gene information core content, we use gene cards and HGNCs. So this will give the comprehensive overview about each gene, like official names, other names, and disease associations, and also connection with the literatures like that. Uh, and also, this helps us to standardize a gene list to avoid confusion from the other naming convention. So the next one is for but core to the protein identification. So one, we have a gene level information. So we have to map again this genes to the their corresponding proteins. So this is crucial because most of the targets are protein structures. So here first we use iniprot. So iniprot is like it is like a very comprehensive resource for the protein data. It has sequences, functional annotations, and domains, motives, etc. in that. Then, so if we want more information about the three-dimensional structure, so we again look at the PDB protein data bank. So which is the repository for the experimental determined protein structure. So very very important databases. So it helps us to see exactly how a protein looks. And for again, protein structure derived, there are many tools for visualization. So that is different. And next, we need to classify the identified proteins. So why we need to classify again? So we have to get the protein structural level information like domains. For that, we use BP from database. And why we use this means because this helps to understand the protein family and the domain architecture. So these are a need for predicting the druggable regions. And also, um additionally, there are also AlphaFold protein structure and Swiss model. So I have told you about PDB. So this is for experimentally solved structures. So for proteins without known structures, we utilize the AI-based AlphaFold prediction 3D structures, so which have revolutionary ability to visualize proteins which lack experimental data. And there are also other online tools like Swiss model, so which is a homology-based modeling to predict these structures and on the related proteins. So from this slide, we can what we get is like each protein sequence, its structure and functional site. So this is helpful to assess if the protein is druggable, and it helps us to proceed with the next step for designing the inhibitor. So the next one is the prioritization of the proteins or the targets. So we have a list of proteins and we have identified the pathways. So we have to prioritize which ones are the best targets. So we use for like filter criteria. We can use essential G proteins, conserved proteins, and the proteins which are absent in humans. So for that, we use network analysis, again, string, gene mania, reactome, cytoscape, David, GSEA. So we can build protein-protein interaction networks and identify the half proteins. And in gene mania, so analyzes the functional and association networks to prioritize the genes. And reactome and cytoscape, so cytoscape is software. So for the network visualization, so this reactome gives us a very detailed pathway annotations. And David and GSEO or the again the gene set enrichment analysis. So and then for the computational target identification, so this is one. It is like an integration or integrated workflow. So we use the BLAST and PSI-BLAST HMMR to identify the sequences. So after all the one, so we have to make sure whether the target is druggable or not. For that, we use docking programs from Schrodinger, Pyrex, Autodog Gold. So this one makes sure that our target protein is druggable or not. So from each one, so I will now show you a demo for it. For example, let me share my screen. So this one is the OMIM TCGA, so gene expression omnibus e.pubmed. So GEA is the one we widely use for the gene expression analysis. So gene expression omnibus. So it is a database under NCDI. So it's a public and functional genomics data, and it is both array and sequence based data. So expression data derived from both microarray and sequence based are available in that. For example, if I give, we have to search using keywords. If we give diabetes, it shows as there are 449,000 data profiles, and we choose data sets. So here left side is the filter panel and center segment is the results, and the right segment is for the organism filter and search details, everything. This is the external link to Geo Home and other one. So on that side, we can see the entry type as data sets, keys, and platforms. These are related to the experimental type, and study type is expression profiling by array or methylation profiling. So we can either choose the expression profiling by array or um expression profiling by high throughput sequencing. So which is for RNA-seq data.
Srimari, how to analyze the data?
Yes, I will again come. So it is another a very big analysis segment. So I will tell you the tools for analysis because it takes more time for analysis. So this is expression profiling, high throughput sequencing. So this is musculus. Okay, just let me go. So here itself we have an option for analysis. We'll check the entry. Okay. So identification of long non-coding RNA. So type 2 expression profiling by Ares, it is associated with human Homo sapiens. So protein expression, gene expression analysis is another set of big analysis. So this is analyzed with GO2R. It's a part of GO1. So using this, we can analyze. So this is another one very big analysis set. So we use this tool to analyze the data from GEO. And so this analysis, by doing this analysis, what we can get is the differentially expressed genes. And also based on different conditions, it can either be drug treated, untreated, or disease and control conditions like diabetes for patients with healthy controls like that. So here T2DM and healthy control. So from these samples, we can get the differentially expressed gene information. And so there, in for gene expression analysis, there are many modules can be used. So GO2R is one which is available online, or else we can use R packages and R tools to analyze. Again, either from the microarray data and sequencing data. So several modules are available like packages. We call it as packages like Lemma, DESIC. So, and we have to normalize a certain statistical analysis to be done. And then at the end, we get the differentially expressed genes. And in case of array express also, we can get the differential gene expression data. And OM, we all know that it is like the genetic diseases, and TCG at last related to the cancer data sets. And in case of gene identification, so gene cards. So from gene cards, there are many other external databases can be connected. So they themselves have like human disease and the annotations and integration of enhancers for target diseases. And this is particularly for non-coding RNA human genes and annotations. So there are more annotation based external links are available in the gene cards. So the human gene database. And the next one is so in the HGNC, we know that Panther, maybe I will go to this in the gene enrichment analysis. So David. So David is again, So when we can use these tools are like once we have the gene list, either from differential expressed genes or from analysis or from literature. So if we know whether the set of genes, what functional data we can derive it, we can use the David tool. So this gene search, or we can either search by a single gene or a list of genes. We can use this David tool for database of annotation. And then enricher is also KEGG is another one widely used for pathway enrichment analysis. So they categorize like metabolism, genetic information, environmental information, cellular processes, human disease, and drug development. So, and also they have overview of metabolic pathways in that. For example, if I take glycolysis, again, this they curate from the experimental data sets. So we have the data. So this, like, if we really know that what our gene occurs, our gene or protein occurs in which pathway, so that we can use the KEGG pathway, so KEGG database. And another one is for network analysis is the cytoscape. So this is one is the way I can string database. So string database, so this is mainly used for protein-protein interaction networks. So we can either by search by name or multiple proteins. So if I give the organism, so protein by name I can give. So for example, if I give EGFR and it is like it itself is the organisms and we have to search. So it again uses the text search and it pulls down the results. So first one is our correct hit EGFR. So that's not EGFR one. So the first one, it is selected and we continue. So here we displays the network, protein-protein interaction network. So again, so this uh, here, yes. So the each color represents different annotations, like from curated databases, experimentally determined gene fusions, co-occurrence, like that. And we can analyze this network here itself. Other or either we can export and we use other external software like Cytoscape and Analysis. So Cytoscape is one software used for the network analysis. Either it can be a gene network or a protein network, and uh, many apps are available in this. So they categorize each app like data visualization, network analysis, and clustering, systems biology, and enrichment analysis. For example, if I choose, okay, top downloaded, it's a Clue Go and code string app. So this Clue Go, what it really, so biological, so it is a Cytoscape plugin, so that visualizes the non-redundant biological terms for a large cluster of genes, which are functionally grouped together. So this clusters the gene according to the functional relationship. So for that, Glugo can be widely used. And string is network, so string app, so that we have seen the string online server, they have app in this. And clusters, mcode is clustering app used to cluster the genes as we discussed earlier, hubs and nodes. And Cluepodia, again, a pathway enrichment analysis for the network. So there are many other apps we can use in this Cytoscape software. And so gene manuals also for functional annotation. it's still loading. And gene virus at escape reactome.
Srimari, I have one doubt.
Yes, yes.
So, how can we study or analyze interaction of food with drug absorption?
Food with drug absorption. Mm, actually, food with I haven't really gone through those concepts between the food and drug absorption. You can post your queries. Maybe next week, sir will come and explain you those queries in that. So far, I have only uh, curated the methods, so not really with the food with the drug absorption. It is more towards the pharmacological effects.
Okay.
Hello, Srimari. Miss Srimari,
Yes.
I have one question.
Um.
You talked about string database today
Mm,
for the protein-protein interaction.
yes. Yes.
A person who is doing PhD, he gave me some 3 to 4 kind of protein name that she got from her, like what, in vitro analysis and everything, from her PhD work, she got it. And now she wants me to frame a string network for
Mm.
it. Once I added those protein names in the string databases, I haven't got nothing. Like it is showing that you are doing some error, you are doing some error. So
Maybe
I'm
you okay in string for single protein you use the protein by name, or if
hmm,
you have a list of proteins, you use multiple proteins, and you
hmm,
have to paste the multiple proteins line by line. For example, EGFR, and below that you have to again type the protein too.
okay, I got it.
So yes, if you have multiple proteins and if you find difficult to paste in this, you
Hmm,
can upload the file. So here the criteria is given like one per line or
hmm.
CSV format. So
O-
then you have to choose the organism.
Okay, okay. And actually, I know how to convert the genes to their respective proteins. But
Um,
can you
uh,
tell me how to convert the proteins to get their respective genes?
a protein, so there you
Genes.
have a list of proteins.
Yeah, I have 5 proteins in that list I have.
Uh,
So I just
either
want to know what kind
you
of
can use gene cards.
gene cards.
Ah, yes. So if I have a protein name, you can itself type the protein name.
Okay,
So it will pull out all the entries in that.
okay, okay. Got
So
it.
we can also get it.
Okay, okay. I will. I will try these 2 things.
So each gene has its reference number according to the
Hmm.
organism,
Hmm.
specific organism specific number.
Okay. Thank you so much.
Maybe I will share you some like simple workouts so that you can easily understand the flow and usage of tools. Like it mostly all online tools and servers or softwares like that.
Yeah, sure.
It's because of the annotation you can remove.
So
These blue lines are there.
yes,
So
yes. Hi.
there's a green annotation tool. Above your mute button, you will see mic button, a green annotation tool. Using this, you can remove this.
Okay. Okay, so maybe I will share the simple assessments
Yeah,
to
you
know
can
more
just
about
give
the tools.
some basic some demo, some disease demo, or some data that you have analyzed, something sort of thing, so that
There's a data for analysis. So I actually it has to start from where maybe I can translate it to the assignments. So I will
no
give the
problem.
step-by-step protocol. I
Yes,
will
yes.
share
I
with them.
think
You need
okay.
data for transcriptomics data analysis. Sorry,
Uh,
for the
uh,
NGS data analysis data, no?
Actually, yeah, yes, we can choose a minimal data for that because it will be computationally intense. So
Yes,
we have to choose data wisely so that we can download in our laptop, also in servers. So
yes,
accordingly, I will share you the data.
yeah, okay.
Because if we do more, I mean, if we choose a very complicated sequences, and then the size will be more, so that we cannot learn from that. So we can learn from very simple data set and use the tools, freeware tools.
Yes.
Okay, so